Hi, my name is Rayaan Shaik, and this is my fall asynchronous presentation for Polygence of 2024. This is my presentation titled Genetic Therapeutics to Treat Alzheimer's Disease. Hope you enjoy. So what is Alzheimer's disease? Alzheimer's is a neurological condition that gradually impairs memory and cognitive functions, leading eventually to difficulty in performing basic daily activities, as well as shortening the lifespan of the individual. An estimated 6.9 million Americans currently live with Alzheimer's, with the majority of the patients being over the age of 75 years old. Some background: Alzheimer's is considered the most common cause of dementia in older adults. Named after Dr. Alois Alzheimer in 1906, he observed alterations in the brain tissue of a woman who had passed away from an uncommon mental disorder at the time. Her symptoms encompassed memory decline, language difficulties, and erratic behavior. Upon postmortem examination of her brain, he identified numerous abnormal clusters, now termed amyloid plaques, and twisted bundles of fibers known as tau tangles, which we'll get into later in this presentation. Just a side note, apolipoprotein E is heavily involved in Alzheimer's. It plays a role in producing a protein crucial for transporting cholesterol and fats in the blood. Disruptions in this function are believed to play a part in Alzheimer's progression. So it exists in various alleles, E2, E3, E4, and having two copies of ApoE E4 is associated with a higher risk of Alzheimer's than having just one copy. But then again, while inheriting this allele increases a person's risk of Alzheimer's, some people with an ApoE E4 allele never actually develop the disease. So it's important to know, but it's not always relevant to the cause of Alzheimer's. We'll start off with our first gene therapy, neurotrophic factor. Neurotrophic factors are essential proteins involved in promoting the growth, survival, and upkeep of neurons. They stimulate the growth of neurons by promoting the extension of axons and dendrites, which are necessary for neurons to connect and communicate with each other. It focuses on alleviating or reducing the secondary impacts of this genetic anomaly by reinforcing synaptic plasticity. While therapeutic strategies targeting tau aggregation, AB142 clearance and growth factor gene delivery, such as brain-derived neurotrophic factor and FGF2, have improved cognitive impairments. They have not halted or reversed the progression of AD. A study involving male mice demonstrates that early AAV delivery of the neurotrophic factor alpha-1 gene to the hippocampus prevented the later onset of cognitive deficits as evaluated by Morris-Water maze and novel object recognition tests. And in the diagram, we can see the nerve growth factor and the BDNF, bind to two families of tyrosine kinase receptors. Also, secreted NF alpha 1 helps protect neurons by interacting with the specific serotonin receptor, which then activates a signaling pathway that boosts the production of BCL2, a protein that supports cell survival. A mutation in the CPE gene found in an Alzheimer's patient has been linked to memory loss and depression-like behavior in genetically modified mice. Delivering the AAV NF alpha 1 gene early to the hippocampus in these male mice helped prevent the progression of Alzheimer's disease by targeting several key factors to restore balance in the brain. Sorry. Additionally, a separate trial was aimed to evaluate the safety and initial effectiveness of using AAV2 BDNF gene therapy to treat early AD and mild cognitive impairment. The therapy involves delivering the BDNF gene directly into the brain using an AAV vector. It involves a one-time gene transfer into the brain using MRI guidance, assessing whether the gene therapy can reduce neuronal loss, stimulate cell function, and promote synapse formation in the targeted brain regions, such as the entorhinal cortex and hippocampus. BDNF, while we're on the topic, is another key growth factor in the brain that helps with nerve growth and development. It also plays an important role in how nerve cells communicate and adapt throughout life. In AD, low levels of BDNF are linked to problems like abnormal tau protein buildup, amyloid beta accumulation, brain inflammation, and cell death. Boosting BDNF, thus, can help reduce tau protein issues and improve cell function by activating its receptor and certain signaling pathways. And in this diagram on the right shows the binding of BDNF to the TRKB receptors, initiating the signaling of several different pathways. The MAPK pathway induces cell growth, and CREB and BDNF gene expression within the nerve nuclei. The second gene therapy is adeno-associated virus therapy, or AAV, like I referred to it as. Gene therapy using AAV vectors has shown great success in treating rare genetic diseases of the central nervous system and sensory disorders. In a study, it was explored whether targeting a specific gene, CD33, with a vector carrying an artificial microRNA could reduce amyloid buildup in a mouse model of AD. And results showed from this study that targeting the CD33 gene, which is linked to Alzheimer's disease, with an AAV vector carrying artificial microRNA can greatly reduce amyloid beta buildup and brain inflammation in a mouse model of Alzheimer's. VHHs, or nanobodies, are small antibody fragments with a single variable domain that can bind specifically to a target antigen. Their small size and unique binding ability enable them to reach specific sites like enzyme active sites that regular antibodies cannot access, offering new treatment possibilities. However, they haven't been widely used in brain treatments because they struggle to cross the blood-brain barrier. Researchers have suggested using a gene transfer method with AAV vectors that can cross the BBB to deliver VHHs directly to the brain. To test this, there was a sole focus on using AAV to deliver VHHs that target BACE1, a protein involved in Alzheimer's. A single dose of this AAV VHH B9, this type of vector, had lasting positive effects, including reducing amyloid buildup, lowering brain inflammation, improving brain function, and enhancing memory for over a year in this Alzheimer's mouse model. Now onto our third and final gene therapy, antisense oligonucleotides. Antisense oligonucleotides, or ASOs, offer a new approach to reduce tau protein levels inside cells, which helps prevent the formation and spread of the tau tangles. Similar to ASOs targeting Huntington, a modified ASO targeting tau RNA led to a dose-dependent decrease in tau levels. The ASO also decreased tau seeding activity in the brain and improved various outcomes, such as preventing hippocampal shrinkage, neuron loss, and extending survival. Researchers are currently developing these drugs. An example is nusinersen, which has been approved for spinal muscular atrophy. This drug not only slows down the disease, but it also improves symptoms in patients who already have the conditions. All right. While there have been some progress with small molecule drugs that affect RNA function, ASOs provide a more targeted approach. Research into antisense drugs for neurodegenerative diseases began around 15 years ago, initially focusing on inherited diseases with clear targets. Early studies targeted diseases affecting more accessible areas of the central nervous system, like the spinal cord. And that strategy has led to one approved drug, 6 drugs in clinical development, and many ongoing research programs. Currently, ASOs are administered through an invasive procedure like a lumbar puncture. This method is generally reserved for more severe, rapidly progressing diseases because it is technically challenging and adds strain on the healthcare system. To make ASO treatments more accessible, researchers are exploring less invasive delivery methods such as subcutaneous catheters. Now in this diagram, ASOs can modulate gene expression through RNase-H1, mediated recognition of an RNA-DNA hybrid followed by degradation of the target RNA. So in conclusion, from these 3 gene therapies and what else we've learned, like from the mouse models, from the studies, everything that we've learned throughout this presentation, basically, research into gene therapy for Alzheimer's disease has shown promise, but there are some significant challenges, some like inconsistencies or stuff that we still want to wrap our minds around. It is one of several emerging treatments that can help slow disease progression or reduce symptoms, though still in its experimental stages. Gene therapy has amassed evidence to support newly found medical approaches for Alzheimer's disease. So may these treatments be very beneficial in the future so that we can slow the progression of the Alzheimer's in many, many patients. Thank you so much for listening to my presentation. I hope you enjoyed it. Thank you.